Hello, everyone. My name is Sumant Kulkarni, and I'm a Managing Director and Senior Biotech Equity Research Analyst at Canaccord Genuity. My current coverage is focused on stocks of neuroscience companies, and I keep track of all the events in the neuro space, regardless of whether it's industry-driven by private, public, or nonprofits, or research coming out of academia. So I'm very pleased to be co-moderating today's panel on advances in neuropsychiatry. I'd like to extend my thanks to our host, Sachs, for inviting us, and to my co-moderator, Manuel López-Figueroa, as well as all of our esteemed panelists and everyone that's tuned into this live video webcast. Today's topic is very relevant as we are seeing a deepening of mental health issues, which has only been made worse by the ongoing pandemic. That said, we're seeing a renewed interest in neuropsychiatry, and several novel approaches are in development. So we remain hopeful that a collective effort will help us tackle these challenging problems that present great unmet need. We'll be keeping this session very interactive, and we'll first start off with a brief introduction from our panelists, and then move on to what I'm sure will be a very lively discussion. If anyone in the audience has any questions, please feel free to send across those using the designated systems interface, and we'll weave those into the discussion. With that, I'll hand it off to my co-chair, Manuel, to introduce himself.
Good morning or good afternoon to everybody. Thank you, Sachs, for the kind invitation to co-share this very exciting panel with Sumant. I'm Manuel López-Figueroa, the scientific liaison of the Pritzker Neuropsychiatric Disorders Research Consortium. And I have the pleasure today to introduce you to the panelists of this, as I say, very exciting panel. First of all, it's going to be Carlos Buesa, who is the co-founder and CEO of Oryzon Genomics. He's going to be followed by David Baker, President and CEO of Vallon Pharmaceuticals. Unfortunately, we're going to be missing Don Wright, who is the President and CEO of Clarigent Health. We're going to have follow-up by Mark Varney, Chief Scientific Officer of PsychoGenics. And the last one, but not the least, Michael Scherz, founder and CEO of Metys Pharmaceuticals. And with that, Carlos, can you please give us a little bit of an introduction to you and Oryzon Genomics, please? Thank you.
Sure, Manuel. Thank you very much. Good morning, good afternoon, everybody. I'm very happy to be here at this panel with this distinguished companionship. So we are at Oryzon Genomics is basically developing innovative drugs in the field of epigenetics in CNS. And I think that these epigenetic components in many of these neuropsychiatric and neurodegenerative disorders is really taking a clear importance in the recent years. So we are working with LSD1. LSD1 is an histone demethylase, lysine-specific demethylase 1. This is an epigenetic target which is involved in many different evolutive processes, and particularly in CNS during development. I'm going to basically to present you a bit more in detail during the discussion the role that LSD1 might do as a key target for personalized therapy in CNS. But we know now that LSD1 is involved also in a number of neurodegenerative or neuropsychiatric disorders. So our company is basically developing this program, and it's actually in phase 2. We are running a phase 2B in borderline personality disorder, which is being run in Europe in 3 different countries and also in the US starting soon. We are also developing or running a phase 2B trial in schizophrenia, scoring cognitive symptoms, negative symptoms and cognitive impairment. And on top of that, and that will be my main discussion, my main participation on the discussion today, we believe that LSD1 might play also a very important role in personalized therapies in rare monogenic syndromes, and in particular, subpopulation of large multifactorial CNS disorders. With that, I will let my next colleague to introduce himself. Thank you very much.
David, if you don't mind.
Great. Thank you, and good morning. My name is David Baker. I'm the president and CEO of Vallon Pharmaceuticals. Vallon's a clinical stage biopharmaceutical company focused on the development of CNS medications with lower abuse potential. We recently completed our IPO earlier this year and began trading on NASDAQ under the ticker VLON. Abuse of prescription ADHD stimulants is a large and growing problem, with data from the NIH reporting that over 5 million Americans misuse or abuse these products annually. And this is especially common with the older immediate release stimulants like Adderall and Ritalin. What surprises most audiences is when they learn that 40% of people who abuse these products report snorting or injecting them. Our lead investigational drug, which we call Adhd, is currently in development for the treatment of ADHD and narcolepsy, and we're leveraging the de-risked 505(b)(2) regulatory pathway. And we designed Adhd using our patented abuse-deterrent formulation technology to be an improvement over Adderall. We've successfully completed our definitive bioequivalence study and a proof of concept abuse study in recreational drug users. We're currently conducting the SEAL study. It's a pivotal intranasal human abuse liability study of Adhd, and we expect this to be the final clinical trial prior to an NDA filing. We're on target for data readout of that study in the second half of 2021 and expect the data to support our NDA filing in the second quarter of next year, 2022. If approved, Adhd is expected to be the first immediate release abuse-deterrent formulation of dextroamphetamine, a drug that's been used clinically for over 50 years and is the active ingredient in medications such as Vyvanse and Adderall. And lastly, our technology is an abuse-deterrent platform that can be applied to a variety of products with abuse potential. We've already initiated work on a second program, an abuse-deterrent formulation of Ritalin, and hope to open an IND on this program by the end of the year. I really appreciate the invitation from the conference organizers and look forward to participating on this panel with my far more impressive colleagues. So thank you very much.
Thank you, David. And Mark, would you mind introducing PsychoGenics? Thank you. You're on mute. You're on mute.
Thank you. So first, I'd like to thank Manuel and Sumant for inviting me on the panel. I'm a PhD neuropharmacologist. I've worked in small molecule drug discovery and clinical development in biotech, mid-sized, and large pharma companies in psychiatry and neurology. I now work at a company called PsychoGenics. PsychoGenics is a company you may be familiar with in that we were one of the early CROs that provided preclinical support to the industry. We have many tens of rodents of models of psychiatry, of neurodegeneration, epilepsy. We have large transgenic colonies, and we provide non-behavioral work in terms of in vitro and in vivo electrophysiology, microdialysis, bioanalysis, EEG, etc. What you might not know about PsychoGenics is we have a second arm, and that is to support drug discovery, and in particular partnered drug discovery programs. We've spent about 15 years. We were really one of the first adoptees of artificial intelligence and phenotypic screening. We have developed high throughput phenotypic screens using mice with artificial intelligence and machine learning. And the idea is to use mice as screens to test unknown compounds and identify specific phenotypes and behavioral changes that occur relative to a reference database of compounds. We've also recently established a similar platform called EQ, which uses EEG to try and map out different pharmacologies. So we've entered a number of shared risk partnerships with pharma, some of which are disclosed, like Sanovian, and others that have not been disclosed. We bring to bear our screening platforms. We can also provide our screening libraries of greater than 5,000 compounds. We can also bring medicinal chemistry. And this approach has really been very successful, resulted in a handful of compounds that are now in clinical trials in the psychiatry and or neurology space, including one that you may have heard about from this conference called SEP856. This is a novel mechanism antipsychotic that's currently in phase 3 clinical trials. And I think what's important about that is it shows it has no activity at dopamine D2 receptors. And of course, you'd all be familiar with the fact that atypical and typical antipsychotics as a primary mechanism interact with the D2 receptor. And here, using phenotypic screening, we've been able to identify a compound with a unique pharmacology. So with that, I'd like to pass it on to my colleagues. Thank you.
Thank you, Mark. And our last panelist is Michael. And Michael, can you please introduce you and Metys Pharmaceuticals? Thank you.
Hi there, my name is Mike Scherz. I'm the founder and CEO of Metys Pharmaceuticals. I'm a chemist, medicinal chemist by training, with a background at Roche and Procter & Gamble Pharmaceuticals, and then many years at Actelion. Metys Pharmaceuticals is developing MP101, NP102, and MP103 for different CNS indications. All three of these molecules are characterized by the unusual, very unusual discovery we've made, that they become pharmacologically more potent when they are present in non-equal ratio of their enantiomers. It's an odd thing that we've discovered and patented. So these patents are granted. It turns out that the parent molecules have existing clinical, preclinical CMC data. And so it's effectively given these compounds a new life. MP101, our lead compound, is a phase 2 ready substance, which we have discovered to characterize the mechanism of its action. It acts as a modulator of glutamate signaling. So many people have developed glutamate signaling molecules. We don't claim to be any. It's not an antagonist of NMDA or of AMPA signaling. It rather a modulator, so it stimulates AMPA type signaling. It inhibits NMDA type signaling in the preparations that we've studied. And that explains that it's also its pharmacology. The compound is active in preclinical and clinical and primate model, rodent and primate models of neuropathic pain. It's also active in depression and in rodent models of cognition. But all of our development efforts are directed towards neuropathic pain, and we're looking for investors to help us finance the phase 2 clinical trials of this molecule in the prevention of chemotherapy-induced peripheral neuropathy, an indication what we think is well suited to the compound's mechanism of action. I think that's enough for the moment, and we'll go on to tackle the panel topics.
Thanks for that intro, and I'll kick it off here. So the way we'll do this is a very vast topic, obviously. So we'll go into some of the main areas of progress and the key opportunities there. Then we'll think about some of the key challenges and how we can all help overcome them. So I'll start off, please keep the questions coming via web interface. I'll start off with a big picture kind of question here. All the panelists went through what their companies are doing. So could you highlight what you consider might be the targets or approaches that might work best, or some of the most exciting new products in development for neuropsychiatry, maybe including your own, or more importantly, excluding your own? So we'll start with Carlos.
I might start. Yes, I think that the main areas of progress in the field are going to be seen once we are able to basically be able to master or to run a more precise molecular medicine. And this is something that has been hampered till now due to the obvious fact that the way that we handle the diseases, the CNS diseases, and the obtention of human samples is quite different than what happens in cancer. So anyway, in spite of that, I think that what we are going to see in the near future is a growing field of precision medicine or precision molecular medicine applied to different subsets of CNS disorders. And this is going to be the basic starting point for a new way that we are going to see on how we conceptualize and run clinical trials and basically aim to cure CNS diseases. And I think that in our case, this is a beautiful example. LSD1 is an epigenetic target that we and others have demonstrated in preclinical models that have an anti-neuroinflammatory effect, that has a neuroprotective effect, that in some animal models we can see changes in behavior, reducing aggression, increasing sociability, enhancing memory. All this is fine, and this is basically driving us to do the classical clinical trials, which are the old concept of all patients in. And this is the way till now the discipline has been advanced with a different target, with a different asset, trying to provide the patients with useful medicine. However, what we see in this screen, and I'm going to try to explain you now, is how the scattered emerging medical evidence can bring us a new picture on how we can understand the molecular basis of personalized therapy in CNS. So what we see here is basically on the left of this screen a collection of rare monogenic syndromes, which are basically caused by monogenic a single mutation that can affect a single gene by deletion, by frameshift, by missense mutation. We have the example of Kabuki syndrome, which is caused by MLL2 loss of function, an histone methyltransferase, a SET1A complex member. This happens one every 30,000 births. And what we know now that this MLL2, which is doing actually the opposite enzymatic action than LSD1, this complex pleiotropic phenotype can be basically rescued by inhibiting the antagonic activity of LSD1. So that has been demonstrated recently by Johns Hopkins researchers using LSD1 inhibitors developed by Takeda. So same thing with the SETD1A, which is another histone methyltransferase, where scientists from Columbia have demonstrated that in adult animals, again, like in the Johns Hopkins case, they were able to revert the complex phenotype produced by the SETD1A mutation by inhibiting LSD1. So we see here like two complexes phenotypes are basically being reverted by LSD1 inhibition. All these that you see on the different shades of green on the screen represent different point mutations belonging to enzymes involved in the methylation and demethylation balance. So we know now that LSD1 can rescue a number of these specific mutations, and this is opening the field for a specific CNS trials on these specific populations. But this is only part of the emerging picture, because we have seen also like SHANK3, which is a protein involved in the glutamatergic signaling between neurons, and is causing the Phelan-McDermid syndrome and a part of the autistic spectrum disorder syndrome. I mean, it's basically also being rescued by LSD1 inhibition. So it's not only confined to this spectrum of histone methyltransferases, and it's certainly going in a more larger amplitude than that. And basically, the fact that LSD1 is able to revert all these different mutations genetically place LSD1 downstream like a molecular hub, where a variety of these neuronal signaling pathways converge. And the fact that LSD1 inhibition can correct distinct independent deficiencies occurring upstream is signaling a way on which we can design new clinical trials. And certainly we can start dissect these large multifactorial CNS disorders, which have been so far very difficult to address in the human clinics, starting by dividing the pie in small pieces. So this kind of divide and impera from divided at impera from the all laughing, I think is going to be also applicable. And if we are able to follow this track, my think is that the main area of the progress of the field in the next years to come will be a progressive conversion on the way of doing clinical trials in CNS to the way we have done these clinical trials on the oncology field in the last 20 years.
Thanks, Carlos. I guess we know your pick. So we'll go to David next, and then Mark and Michael.
Sure. So I'll, for the time being, kind of talk about areas that excite or maybe better intrigue me outside of the work that we're doing in abuse deterrence. I think closer to home, closer to more mainstream is the emergence of some of the fast-acting antidepressants. I think that's a very nice development, and I think we're going to see more of that in the coming years. I think as you start to move away from there into, perhaps a little less traditional, more novel, I'm intrigued by the work with the endocannabinoids beyond CBD. We heard, Harry Tracy talk a few moments ago about the psychedelics. I think it's an interesting area. I'm not sure I'd say I'm excited about it yet. I'm reserving judgment, but I'm intrigued by it. And then, outside of our mainstream areas, I'm excited to see what's going to end up happening in the real world effectiveness of digital therapeutics. But also, it may sound unusual from someone who's trying to develop drugs and trying to develop improvements on existing drugs, but I'm very intrigued by what's emerged over the last few years in neurostimulation, whether it's trigeminal nerve stimulation, where we've seen, some good work in our field, ADHD, or repetitive neurostimulation in depression, which seems to work. It just needs to get paid for. I just think there are a lot of opportunities there that really go beyond kind of our traditional pharmaceutical therapeutics.
That's great, David. Thank you.
Mark?
Yes. So I think one of the things that's quite interesting is that in mental illness, I think we're much further behind with neurodevelopment disorders and neurological disorders in terms of identifying new targets for the treatment of depression, anxiety, and schizophrenia. And that is in part why I'm really so supportive of phenotypic screening. But I think what I find interesting is kind of a resurgence of old targets in the psychiatry space. And so, for example, and again, this came up earlier with Harry Tracy, Karuna's M1, M4 agonists for xanomeline with their peripheral antagonist, I think would represent a potential new mechanism for the treatment of schizophrenia. I think it's very exciting. But that was a discovery that was made 20 years ago, and just some clever ideas about how to reduce the side effects. I think the same can be said for the psychedelics. In contrast to David, I'm actually pretty excited with the psychedelics. I think there's an opportunity for not only rapid-acting mechanisms, but also long-lasting mechanisms that may provide longer-term treatment, similar to ketamine. And of course, those target serotonin receptors, which were in vogue in the 90s. And sort of we think about, we've been there, we've seen it, we've done it, and here we are again, back at 5-HT receptors. And I think part of that, particularly with the psychedelics, one of the main issues is going to be overcoming any of the dissociative or the psychedelic activities. Can we actually separate out the medicinal aspects of the molecules and remove the psychedelic components? And I think there's some data out in the literature now, and from some of the companies that are working on this suggest that you can do that. And I think while we don't understand the mechanisms, an area that I've worked in the past, I think is really interesting is in biased agonists, and the fact that not all agonists that activate a receptor do so in the same way. And some of these compounds can activate the receptor in unusual ways and activate non-canonical signaling events, which may actually have quite a different readout in the animal. And I think this may well be the case with the 5-HT2A receptor activation and the fact that not all 5-HT2A agonists produce psychedelic behaviors in animals. So that's an area that I'm personally watching very closely. And then finally, one other area that I've kind of see that I think is interesting is really the sort of conjoining of the neurology and the psychiatry fields in CNS disorders. And I think, while they've been separated based on clinical indications, what we're seeing now is the fact of BPSD, behavioral and psychological symptoms associated with dementia. You're now seeing the recognition of the importance of some of the psychiatric behaviors that occur in Alzheimer's, in Parkinson's, and Huntington's disease. I'll leave it at that.
Mark, while we have you, I'll just give you a quick question from the audience, 30 seconds on this. What's the throughput of your phenotypic screening platform with mice? How many compounds can you screen, and is it a whole animal screening model?
The whole animal screen, the test takes about 45 minutes. We have, it's scalable, so we currently have about 35 chambers that are set up that are run up to about 8 times a day. And we can screen in the range of tens of compounds per day. So, up to maybe 100 a week or so. That is scalable. So in some instances, we have gone to 2 or 3 shifts per day when required, if we need to go to additional compounds. It is not high throughput in the terms of in vitro 384 well screening, but we get so much information and surprisingly quite a big hit rate of about 30% of compounds that we look at, compounds that are curated from a chemical library, we see produce some behavioral effect.
Got it. And then we'll move on to Michael. And Michael, when you're addressing this, there's also a question from the audience.
I've seen it
specifically.
Yep.
So we can do that. Just before that, I have a quick disclosure to make. Canaccord Genuity on the investment banking side has performed services for Compass and Cybin on the psychedelic side. And so the mechanism mentioned, I also have to mention intracellular therapies. With that, I'll turn it to Michael, please.
So the question, as I understand it, is what other therapies are interesting that you're pursuing that make sense in neuropsychiatric indications? And I'm a reasonably well-read medicinal chemist. It's just to be a long list, but some things have really caught my eye because of where they're being used. They act, however, in the periphery. I believe they act in the periphery. These are HDAC6 inhibitors that have been of interest. These HDAC inhibitors have been interested in chemotherapy for some time. But there's been a body of work that's shown that these HDAC6 inhibitors specifically involve or specifically accelerate the remyelination of damaged peripheral nerves. Well, when you do that, it actually reduces the symptoms centrally, right? Not because the drug is acting in the brain, it's acting in the periphery, but the drug effect becomes apparent in the CNS, in the symptoms that the patient experiences. So these HDAC6 inhibitors are also being studied for chemotherapy-induced peripheral neuropathy for the damage, direct physical damage done to the peripheral nerve. Our molecules act differently. Our molecules do not affect the peripheral nervous system. They have an effect on higher brain function, depression, cognition. But those effects we don't ascribe to the action of the drug in the spinal cord. In neuropathic pain, though, our molecules' actions can be understood in terms of the effect that they're having on the only synaptic connection that the damaged peripheral nerve makes to the central nervous system. In the spinal horn, the dorsal spinal horn of the spinal cord, that synaptic connection involves glutamate for the synaptic strength. There's a learning mechanism when the peripheral nerve is damaged, and that synaptic strength increases and increases, and that sensitization phenomenon is driven, there are books on this, by glutamate signaling. So that's how we understand our drug is working, not to prevent the damage that's being done to the peripheral nerve or repairing the damage to the peripheral nerve, but rather interfering with the signaling pathway of that peripheral damage that must, of course, at some point enter the CNS. So our compounds act as modulators of glutamate signaling. By that we mean they are not channel blockers. So glutamate normally opens a cationic ion channel, and ketamine, MK1, adamantamine, other compounds, they block that channel irreversible in a use-dependent way, but it's a block. The channel is turned off. This has toxicological consequences that are significant. It's one of the reasons that people have been looking for modulators, molecules that act in different one says allosteric sites. That means basically we don't know exactly where they're acting, but they change the way the channel functions. There are crystal structures that we've become very interested in of a molecule called piracetam binding to a glutamate-gated ion channel. And you can see that the piracetam molecules, plural, change the way the glutamate binding domain works. There are, in fact, 6 molecules of piracetam in that crystal structure. Our molecule, dimiracetam, is a rigid analog of piracetam. We suspect that our molecule is binding to the same sites, not in the channel, but in the torus around that channel. And interestingly, our molecule, chiral molecule, only 2 of those 3 unique binding sites would fit our R enantiomer. The 3rd site does not fit the R enantiomer, only the S site. So it becomes a way of understanding the molecular origin of this preference for a non-racemic mixture. On AMPA signaling, there's an increase in current. On NMDA type signaling, there's a decrease in current, but not by blocking the channel, but instead by modulating the way that those proteins, those large complicated proteins, function. I hope that addresses the questions been raised. Oh.
Thanks, Michael.
All right. So if you don't mind, I'm going to go into the next topic of discussion today. And we cannot obviate that over the last year, we all have been affected by COVID-19. And if you listen to what Harry Tracy presented earlier, about $2.2 billion have been invested in CNS. About 30% of that has been in psychiatry, and about 50% have been in M&A activity in the psychiatry space, according to Harry. However, as we all well know, mental health has been highlighted in the last year. The increase of psychiatric disorders has been unprecedented, and also the way that COVID have affected the members of the panel in either conducting early stage and research and development in their clinical development, or how they started to apply digital health have had an impact on their companies. So our next question relates to, could you please characterize how COVID have impacted your, again, whether it is clinical development, research, or in the case of Mark, how actually that have been beneficial to the companies who are working on a more virtual form. So why don't we start with Carlos? If you can briefly, please give us your feedback on this topic. Thank you.
Well, thank you, Manuel. I think that it's obvious that this new situation, this pandemic, has affected us all. And I think that we need to be honest. I mean, we have done our best to minimize the interference and the damage and to maximize the emerging opportunities. But it is true that in general, when you run clinical trials on the early months of the pandemic, the situation was quite critical. So a number of trials were recruiting very slowly because of obvious reasons. Now that we are facing the end, what we are facing is a backlog of work that has not been done during this past month in the hospitals in the site, which is going also to have to be digested by the clinical institutions, by the sites and by the CROs. So that's going to produce a sort of deferred state on the starting clinical trials. So we are facing that. We are trying to get remedies to that, and we are trying to cope with that on the best way we can. Also, the way that we have been dealing with partners to try to identify alliances and to investors to be able to communicate the promise of our programs has been affected. I mean, the proof of that is that we are in this very lively panel, but we will not enjoy the after panel that we would normally have done if we would have been all of us in Zurich or in Basel. So this kind of new ecosystem of interaction with investors and with partners have certainly some upsides, because you can have many meetings without the hassle to travel and the time zones and so on, but has the downside that you reduce the serendipity, the human touch, and so on. And that's also affecting, specifically when you have early programs in which it is not only the quantitative data, but the qualitative touch that you can provide in a face-to-face meeting. So big challenge, also some opportunities. I think that in general, what is also clear is that the financial ecosystem has been not particularly hostile to the sector, and that's good. And we are also appreciating a general interest in CNS. Oryzon is a dual company. We are working on oncology, but also in CNS. We believe that our most distinct and novel approach is in CNS, where we are, proposing this personalized medicine approach. And our take here in the last month is that, yes, we have got a substantial manifestations of interest. So we are hopefully cautiously optimistic, but certainly hoping that we can get back to normal, because I don't think that this sort of new normal is the optimal one. We have done the best we can do, but that would be my take on that.
David? Thank you.
Sure. So I think there are a few ways of looking at it. You can look at the way that COVID has impacted our industry and our execution more broadly. And then I think there's the more specific effect because we work in CNS and the impact of COVID on CNS and mental health. Speaking for us, in some ways, operationally, we operated in a very virtual manner before COVID, so that has not been overly disruptive. There have been disruptions with some of our key partners, CROs, manufacturers, and so while it hasn't been terrible, we've had to, there've been some delays and we've had to play catch up. Fortunately, we're back on track with everything. I think one of the positives that's coming out from this is that I think COVID reduced some of the resistance and some of the friction towards a move towards telemedicine and telepsychiatry, both clinically and I think in terms of execution of clinical trials. And I think, now that resistance has been removed, I don't see that going back, certainly not entirely. And I think that's good for clinical treatment where there's a real need for telehealth with mental health, and I think it's going to be helpful with clinical trials. That said, for us, our big focus right now is conducting human abuse liability trials. Unfortunately, they are not particularly amenable to doing at home with telehealth visits. You have to literally bring in our case, recreational drug users and expose them to controlled substances, not something really one can do from home. But all in all, I think the trends towards better telemedicine is a good thing. I think for us, we've managed to manage through it and feel that we're in good shape progressing further. But for the broader field, I think they'll net be a positive effect.
Thank you, David. Mark, I would be curious to hear what PsychoGenics is going through for COVID.
Yeah, so on the we've sort of been impacted in multiple ways. I think on the CRO side, we really, due to the foresight of our CEO, Ema Leahy, we set up a PCR COVID test in the labs and monitored people on a frequent basis. This is back in March 2020. When you couldn't get tests. So we were actually able to, in doing that, we were able to actually keep the labs open. And I think as a result of that, we did see an increase in work because a lot of other labs were closing down. So from the CRO side, I think actually everything was fine. It just required us to constantly monitor the staff that were coming in, isolate people if they were positive, decontaminate, the work areas, etc. And so we've been pretty successful. We, on the drug discovery collaboration side, we were affected. We do our chemistry offshore, China and India, and there were periods of time where those labs were shut down, which impacted our drug discovery programs. And then I sit on the board of a neuroscience company who were trying to start a phase 2 study in Europe in an aged population, and that all got delayed. I mean, it was proving to be very difficult to screen patients appropriately to enter the trial, and so we just sort of have pushed that back and hope to push that up. Hope to start that towards the end of the summer. So yeah, it's sort of been impacted in various ways.
Cool. And Michael?
We don't have any ongoing clinical trials, and so we only saw the effects in the financing side, where one venture capitalist who submitted a term sheet had to withdraw the term sheet when his Chinese funding source dried up with COVID. There's also a Chinese company who had entered due diligence procedures and also had to withdraw. So there was a very direct effect on us on the financing side, which so far have not recovered. We found other sources of funding, and I'll come back to say more about the governmental funding that became available through the city of Basel. I'll come back to that. The other area where we certainly saw a very immediate impact was in the quotes that we had from CROs and manufacturers of clinical trial supplies. Those quotes became worthless when it was clear that they were having closed facilities manage their project flow slowly, and also the helpful change in the rate of telemedicine that clearly has reduced some of the costs from the quotes that we had for the CRO operations. Then a quick word about what the city of Basel have done here in Switzerland. Very unique in Switzerland, I think even in Europe. A program was put in place by the government of Basel to provide emergency financing for companies who could demonstrate an effect of the COVID situation on their operations as startup companies. So these were startup companies in healthcare in the city of Basel, were invited to submit a description of the problems they had encountered and what the impact had been on their business. We received a grant for 1.8 million francs to make up for the damage from the loss of those two Chinese connections that were due to COVID. I thought it was a very forward-looking and program, and it was implemented with remarkable alacrity and an absence of bureaucracy. It was a beautiful thing to behold, and I was quite impressed. I think a lot of other Swiss cantons also watched very carefully what had happened here in Basel. So, having said that, we eventually had to turn down that grant, but that was for us an interesting experience.
Okay, thank you, Michael. So let's move to the next topic, which I'm a big believer on, which is personalized medicine. And in particular in psychiatry, we have seen in the last year a number of developments that are very exciting and is allowing us to move in that direction. I'm talking about, for example, the use of imaging like functional MRI and looking at the connectome to identify different subgroups in patients with psychiatric disorders. I'm talking about the implementation of digital health like CBT as ways of treating specific subpopulations, or the use of biomarkers, whether it is EEG or blood biomarkers that is allowing us to identify those clinical subphenotypes. And in the panel, as you can see, we have several companies that are applying those. And I would love to hear from each of you, where we are and where we're heading when it comes to, personalized medicine in particular. And maybe start with Carlos, which already gave us a little bit of that background, but maybe you can have another minute to enlighten us on the topic. Thank you.
Glad to do that, Manuel. No, thank you. I think that, as I said before, one of the things or the aspect that have hampered the progression of molecular medicine or personalized medicine in CNS is obviously the way that we handle the disease and the obtention of specimens and human tissues and human samples. So we have tried for decades to get surrogative markers, peripheral markers of the status of the disease with, I would say, not particular success till now, at least in the Alzheimer's field, as we have seen. It is difficult to really dissect biomarker from being a sort of consequence or a causative. So the etiology, the molecular etiology of these diseases are still basically the major caveat for us to advance into the real personalized medicine concept. So I think that what we are seeing as a matter of major advancements are these kind of genome-wide analysis, all these genetic correlations, which are basically starting to put the spot on specific areas. And I think that the epigenetics is also going to provide an additional light on how these wrong transcriptional programs might affect different neuronal subtypes to produce what we are identifying macroscopically as a certain disease. So we might find, and I think we will find in the future, that most of these large multifactorial CNS disorders will be split in a number of different genetically defined, like it has happened in the oncology. Right now, it's absolutely normal for us to understand that we can deal with 10 or 15 different of breast cancer or 10 or 15 different types of lung cancer. So I think it's going to be very similar situation on the large CNS disorders. And I think that these monogenic conditions are going to provide us the tool, especially now that we are starting to have the clinical assets which are able to basically validate or invalidate the hypothesis. So once you get a clinical asset which is brain penetrant, which is active at the nanomolar level, which is producing very low or level of side effects, you have the right safety profile, you are in this position to really basically start validating this hypothesis if certain monogenic disorders can be reverted. In the example I was providing was the lazy one, but I'm sure that in the future there are going to be many, many other examples. So, and that might also start opening the way that we deal with the large disorders. So I think this is basically going the way to go. I'm a bit more skeptical about the imaging and the electrophysiology. I think that this is something that we need to keep doing, but I am not sure that we are yet to the technical point that is going to provide real, black and white answers to a number of the big questions in these diseases. So this is my take. My take is going to be a long way, starting by the genetic correlation, monogenic disorders, and advanced specific and selective drugs that can validate those hypotheses.
I'm not necessarily agree with your last point, Carlos, but I think that there have been really exciting advances in the use of imaging EEG in the parceling of populations. But I'm going to let David comment and have another question similar.
Yeah, and I'll take my
Let's be polemic.
Yeah. I'm going to jump in between the two of you, which is to say, I don't think we're there yet in terms of imaging, but I do think we've made progress. And I can tell you when I first got involved in CNS and in ADHD in particular, we had to spend a lot of time trying to dampen unrealistic enthusiasm and beliefs that many patients had that imaging could help with diagnosis and specificity of a condition like ADHD. And at the time, we would, on the one hand, say, look at this population-based imaging data, but it doesn't necessarily translate and can't be applied at the individual level. And if you had asked me 8 to 10 years ago, I would have said, we're not going to see that during my career, maybe not in my lifetime. I think the imaging has improved and advanced. I'm not sure that we're there yet, but I've seen major improvements. And so I'm optimistic that the field will continue to advance there. I think the prospect of using biomarkers as well for parsing out patients is very promising. It has not been as relevant in some of the fields that we have focused on, such as ADHD. On the other hand, just kind of a cautionary note, I look at a field like Fragile X syndrome, which I've looked at a lot and been involved in clinical work and paid very close attention in that field. And frankly, it's been fairly disappointing. And now there's talk that there's much more heterogeneity in a population that we would have thought is much more homogeneous. And so I think this is the direction we're going in, but I think we're going to have to go there, and I think we're seeing improvements, but there still are some cautionary tales that at the end of the day, when you run a trial in what is a reasonably heterogeneous population with genetic mutations, and you're not seeing good results, you have to kind of say the answer may not be just parsing the population further. So I'll leave it at that.
I just want to highlight, among others, the work of Atkin at Stanford using EEG, or the work at Cornell with Connor using functional MRI, which is showing, as you say, really interesting data on how to parse the different populations. But let me ask Mark, you guys have been a pioneer on the applications of AI, artificial intelligence. And, as we're gathering more information as it relates to not only molecular phenotypic behavior, but also clinical, and people are trying to integrate those. Are we there yet? Or, are we, as David is saying, not in our lifetime? I mean, where we are in psychiatry with this, and you're immune, Mark, if you can unmute yourself.
Thank you. So we actually have a lot of interest from clients who want to do rodent EEG as a potential biomarker for their phase one's kind of target engagement. And that's part of the work that we've been doing with the EEG Cube, which is to basically take I think a couple of hundred reference compounds, CNS reference compounds, and to evaluate them at multiple doses in a mouse and look at EEG changes that occur. The challenge that we've had is just purely the amount of data that you get and the way that you have to analyze that data. And so artificial intelligence is a big component of that analysis. I would say that we have worked our way through a lot of that. We have classifiers now that seem to be quite accurate. The question is, can you then potentially translate that to the humans? And I think there are more challenges there. Obviously, the rodent brains are smaller, and you get only a specific depth of measurement with those electrodes. And since the human brain is so much larger, then obviously you're only measuring really surface activity with the EEG relative to rodents. So I think there are some fundamental challenges. We're also on our drug discovery programs. We like fMRI and CFOS as measures to look at brain activation, and of course the fMRI can translate to the clinic again for target engagement. And then the other aspect of EEG that we haven't talked about is sleep. Looking at changes in sleep. It's, sleep is a very easy measure, particularly changes in sleep architecture is a very easy technique to use in your phase one studies. And of course, in animals, we've generated a lot of data there. And I mentioned early on in the introduction about this compound CEP856, this novel antipsychotic. One of the first studies that was done in humans to try and get since the mechanism was not clear at the time, we couldn't use conventional PET type ligands or other biomarkers. But one of the things we noticed in animals is a profound suppression in REM during sleep. And so this was actually used as a biomarker in the phase one studies. Not particularly innovative, but did identify doses which were able to do that. And subsequently, those doses ended up in phase two studies being efficacious. So I think EEG is very valuable, both from the sleep and the non-sleep perspective. But sometimes there are other techniques that we have to engage to support those studies.
Okay, thank you, Mark. Very exciting. And Michael, we're almost running out of time. So can you, in the context of personalized medicine, can you comment on whether,
I can be very brief. In our main phase 2 clinical trial for prevention of oxaliplatin-induced peripheral neuropathy, we make no effort to introduce concepts of personalized medicine. 90% of these subjects experience a cold sensitivity. That's our primary endpoint. We have no reason to expect that there'll be major predictive outcomes that would change that. So in that study, nil. However, in painful diabetic peripheral neuropathy, for several reasons, including this question of personalized medicine, we have designed the trial as a randomized withdrawal design, where patients first go on 5 weeks of open label treatment with MP101. If they do not respond, they discontinue. They leave the study. If they do respond, we'll keep them in the study, but randomize them to continue on the active drug or placebo under double-blind placebo-controlled conditions. So that is, in fact, a personalization strategy, and one that I think has a lot of merit to it, especially where there are existing therapies for that same indication. There's a third area where personalized medicine is a potential. It's again in this oxaliplatin-induced field, in a monkey model of oxaliplatin-induced peripheral neuropathy, where we can prevent the cold sensitivity in these primates. We can also prevent the functional MRI signal that's been associated with that cold sensitivity. So there's a functional MRI correlate, and it's a beautiful, it's like a thumb sticking out of your brain. It's a very clear, specific bilateral signal, which goes away with treatment with MP101. At the same time, the cold sensitivity goes away. So I think that's a very interesting thing. But my God, the price of running a functional MRI compared to asking a patient to score on a scale of 0 to 10, the cold sensitivity in the last 24 hours, it's not comparable. So it's an interesting thing. It's curious. It may turn out at some point to be useful, this functional MRI component. But by and large, we've been using classic drug development techniques and clinical trial designs in order to get to this.
Sumant.
Yep, thanks for that. So we have a minute left, and I'm asking everyone to keep this last kind of answer very tight, buzzer beater type. We'll move on to the financing environment in neuropsychiatry. So what could relatively small or young organizations like yourselves do differently to attract investment for these types of diseases? And there are some exceptions, but what do you think it'll take to get major biopharma interested in neuropsychiatry again?
I think biopharma is interested in neuropsychiatric indications, and big pharma are interested in neuropsychiatric indications. We've so far been unable to secure venture capital financing. We have received an offer now from a publicly traded biopharmaceutical company to take over Metys. But I have to admit, I've been disappointed with the kind of interest that the venture capital community have shown for our new composition of matter patents for existing drugs. I don't get it. They're distracted by new treatment modalities. They're distracted by I'm not sure what. No, I'd have to say it's been difficult here in Europe.
I would just say that fortunately, the last year has shown not only is there continue to be big pharma and medium pharma interest, but the public market opportunity has just completely changed around. And I think for just an example, I wouldn't have anticipated we would do it, not looking to raise a lot of money. We were looking to raise money privately, but we were able to go out in the middle of COVID and do roadshow by Zoom and phone and oversubscribe and take ourselves public and secure our financial future for the next few years. So I'm actually very pleased and optimistic about what we saw and what I would expect others can see. And I think the fact that financing environment, at least on the public side, has been encouraging, allows small companies like ours to advance ourselves to the point that we will get the more established companies to be more interested in us. At least we're not, we're able to get the funds to do that work to the point that they're going to be interested.
Can I just make one comment?
I'm afraid that they already closed the session. So as much as this is a very exciting topic, and of course, we want to thank you all for your participation in the panel, and all your participation have been extremely helpful. So, Sumant, will you close and thanks again?
Thank you, Manuel. Thank you, Manuel. And
thank you, everyone, and hope we can all meet in person at some point soon.
Gladly, yeah.
Very interesting. Thank you for the thought you guys put into organizing the panel. Very interesting. Could have talked about this for at least another hour, but
at least.
Yeah. I'm very interested, Manuel, to hear more about your functional MRI work. I'll be looking up some papers soon. And maybe drop you a line as well.
Absolutely. Thank you.
Very good. Thank you, everyone.